This is Dr. Ahmad, and today we are going to start a new presentation. We are going to start a new discussion. It is going to be on basal ganglia. Today we are going to discuss the structures, connections, direct and indirect ganglia pathway. We're also going to look into Parkinson disease and Huntington disease, so we have a better understanding of how basal ganglia controls them. So, basal ganglia are basically a group of subcortical nuclear agglomeration which is involved in the movement and are located deep to the cerebral hemispheres. And there are the cluster of nuclei, which are made up of the subcortical nuclei, and they're involved in initiation, maintenance, and inhibition of the movements. So there are four major structures of basal ganglia, which are further divided into further areas. We have striatum, we have the lenticular nucleus, substantia nigra, and subthalamic nucleus. In this image that you see here on the right is the cross section of the brain from the anterior perspective. You're looking at it. You see the thalamus right here. These are the cerebral hemispheres. There's a thalamus in the middle right there. There's putamen. We have a subthalamic nucleus. Substantia nigra, we have red nucleus right there. We'll discuss red nucleus in the cerebellum. There is the cerebral peduncle. We have caudate nucleus, the tail of it. We have globus pallidus, the internal segment. Then we have the internal capsule, which we have already discussed. We have the globus pallidus, the external segment. We're going to look at that later. And then we have the caudate nucleus. So the first area of the basal ganglia is the striatum, and it is made up of further two parts. We have the caudate nucleus and we have the putamen. The caudate nucleus is an elongated C-shaped structure which is divided into head, tail, and body. It is running adjacent to the lateral ventricle, and the head of the caudate nucleus is continuous with the putamen. The tail tapers and it terminates near amygdala in the temporal lobe. The second part is the putamen. The putamen is a large nucleus which makes the lateral portion of the basal ganglia. It fuses with the head of the caudate nucleus anteriorly and ventrally. The second portion of the basal ganglia is the lenticular nucleus. The lenticular nucleus has two parts. One is the globus pallidus, which is a smaller triangular structure consisting of numerous myelinated fibers, and it has the internal and external segment which acquires input from putamen and caudate nucleus and communicate with subthalamic nucleus. And the second portion of the lenticular nucleus is the putamen in itself, which again is a large convex gray mass, and it lies lateral and inferior to the insular cortex. We have putamen here in the striatum, and it has the portion with the lenticular nucleus as well. The third portion to the basal ganglia is the substantia nigra. It has two portions, ventral and dorsal, and the degradation of the neurons here in substantia nigra is important in the pathogenesis for the Parkinson's disease. So the ventral portion is called the substantia nigra pars reticulata, and the dorsal portion is called the substantia nigra pars compacta, and that is darkly pigmented dopaminergic neurons. And the final part of the basal ganglia is the subthalamic nucleus, which lies under the thalamus and is spindle or cigar shaped nucleus, and it derives from midbrain. And unlike the rest of the thalamus, it is derived from the forebrain. So here again, we have the lateral view of the basal ganglia. Again, we have the same image that we have seen the anterior view. You see globus pallidus is underneath the putamen right there. We have the head of the caudate nucleus. It goes above and it tapers into the amygdala nucleus. We have the lateral geniculate body, which we have already discussed in the previous lectures. This is the tail of the caudate nucleus. We have an optic tract that go here, and the putamen is right there. Again, this is the anterior view. You see the thalamus that is like right in the middle of everything. Red nucleus, we're going to discuss that with cerebellum, and we have substantia nigra, which has two parts in itself. We have the subthalamic nucleus. So starting from outwards to the inwards, we have putamen. We have the caudate nucleus. We have caudate nucleus, putamen, and then we have the globus pallidus, internal and external segments in itself. Again, then we have the caudate nucleus right there. So what are the basic functions of the basal ganglia? The basal ganglia has a significant role in controlling the posture and the movements. It also has the connection with the limbic system pathway, and it governs the expression of the motivational states and behaviors. It also has a major decision making link in the brain, and it controls the eye movements as well. So we're going to look into the pathways for the basal ganglia. We have the neural pathways, and then we have the intrinsic relations. So let's start with the neural pathways first. So it has two pathways, the direct pathway and the indirect pathway. The direct pathway is excitatory, so it is going to have a positive influence. So basically, this signal starts initiate in striatum. It reaches the globus pallidus internal segment. This is important. Then we have thalamus that gets involved. Then we have the cerebral cortex, which finally gives feedback back to striatum. So the whole cycle, the whole pathway starts with striatum, ending in striatum, but the feedback is positive. So hence it has a positive influence. The second one is the indirect pathway. The indirect pathway is inhibitory in its function because it is going to cause the negative influence. So again, it starts with striatum, but this time instead of going to globus pallidus internal, it goes to globus pallidus external. Subthalamic nucleus is going to get involved. In the previous one, we see that the thalamus got involved with that, but in this one is going to involve the subthalamic nucleus, which then gets involved the globus pallidus internal. So both nucleuses of the globus pallidus, or both segments of the globus pallidus, are going to get involved. Now again, the after the globus pallidus internal, it will activate the thalamus, then the cerebral cortex, and the negative feedback goes back to the striatum. The third neural pathway is the nigrostriatal, which is going to start in substantia nigra, and it feeds directly to striatum. And this pathway directly modulates the direct and indirect pathways. The basal ganglia then have the intrinsic relation. So there's two type of intrinsic relation. One is inhibitory, and the second one is excitatory. The inhibitory pathway is GABAergic, and it consists of striatopallidal, striatonigral, and globus pallidus external, which is inhibitory to the subthalamus. And then there's an excitatory, which is glutamatergic and dopaminergic both. So it has only one relation, the nigrostriatal, which is dopaminergic. Now we're going to start the Parkinson disease. After that, we're going to look into the Huntington disease. So first of all, the definition of the Parkinson disease. This is a clinical correlation, so you have a better understanding when you further look into the pathology. So Parkinson disease is a chronic progressive neurodegenerative disorder with the affected patients presenting with resting tremors, bradykinesia, rigidity, and postural instability. The risk factor involves the environmental and non-genetic risk factors, which includes the exposure to pesticides, nitrogen dioxide, traumatic brain injury, rural environment, drinking well water, and the use of MPTP, which is 1-methyl 4-phenyl 1,2,3,6 tetrahydropyridine. I know it is mouthful. Then we have diabetes mellitus type 2, which is also one of the risk factors. And then we are also going to look at the gene mutation, the alpha synuclein SNCA gene, Parkin2, Park2, as Parkinson, so Park2 gene mutation, etc. What is the pathophysiology? So it all starts with the depletion of the dopaminergic neurons in the substantia nigra pars compacta, which will then progress into the depletion of the dopamine in the nigrostriatal pathway, which then further develops into the motor symptoms. And this all combines with the overactivity of the indirect pathway. If you remember, indirect pathway is basically going to influence the negative feedback. So overactivity of the indirect pathway functionally disables the functioning of the substantia nigra. Then what happens? So initially there is a compensatory mechanism in the brain. So temporarily the direct and indirect pathway, it will try to balance itself out. However, it may temporarily decrease the effects of the dopamine depletion until these mechanisms are overpowered by the disease progression. There are six stages of the Parkinson disease, which is called as the Braak staging. Stage 1 and 2, there is a changes in the olfactory bulb and medulla oblongata. Then we have the stage 3 and 4, where the symptoms, when the pathology migrates to substantia nigra, pars compacta, and then the other structures in midbrain. And the stage 5th and 6th, which is the last stage, it is the pathological process that reaches the temporal lobe and the frontal lobe. And the symptoms include bradykinesia, tremor, which is the pill rolling movements. And these tremors, they decreases with voluntary actions. This is important to remember. Then we have the rigidity, postural instability, speech impairment, dysphagia, stooped posture, with other symptoms as well. So the last one, dysphagia and stooped posture, is one of the minor symptoms, but the other ones are the major symptoms. The structural changes. So histologically speaking, we have Lewy bodies, which are round, eosinophilic, intracytoplasmic, neuronal inclusions, which are seen in substantia nigra, locus coeruleus, cerebral cortex, and sympathetic ganglia. And then there is the depigmentation of the substantia nigra. We're going to start the Huntington disease. It is also a clinical correlation with the basal ganglia. Again, it is a progressive neurodegenerative disorder, and it has a genetic component of the autosomal dominant mode of inheritance and poor prognosis. It is caused by the autosomal dominant genetic alteration of the Huntington gene, HTT, on chromosome number 4p, and there is an expansion of the CAG. CAG are the nucleotides, so these are the cytosine, adenine, and guanine. So they repeat numbers over and over in the successive generation. So in these successive generations, you're going to see that Huntington disease presents earlier and it becomes more severe. So mechanism of anticipation is unique. So the normal CAG on repeats on chromosome number 4 are usually 27 or less. But in the Huntington gene, the threshold for developing this disease is the 36 repeats. So this is an important high yield point. In the chromosome number 4, if it is the CAG repetition is 27 or less, it is normal. But in the HTT gene, the development for the CAG, or the expression of the CAG, is 36 or more. So the length with these repetition, it correlates with the severity of the disease symptoms. So what happens here? So there is a toxic gain of functional mechanism. The mutant gene, it gains deleterious function, and these abnormal HTT protein fragments, it undergoes misfolding and aggregation, which will increase the fibrillar aggregates, the abnormal proteins, which then form the neuronal inclusion bodies. So the neurodegeneration begins in the caudate nucleus. In Parkinson disease, the stage 1 starts with the olfactory bulb, but here in the Huntington disease, the neurodegeneration begins in the caudate nucleus, putamen of the basal ganglia, and the cortex. So eventually it involves the entire brain, and then it caused finally the neuronal death. What is the pathophysiology? So up till now, we have discussed how the genetics works into this, but how it takes part in the major parts of the brain. So the major component is the basal ganglia normally. So there is an equilibrium between the direct and the indirect pathway, again to facilitate the normal motor movement. So the body or the brain decides to maintain the balance in both direct and indirect pathway. So what happened here is that after it starts aggravating, the disease starts to progress. The direct pathway, there is a dopamine problem. We have the excitatory connections with thalamic nucleus. So this is basically the normal function, how the direct pathway starts. So there is a dopamine which causes the excitatory connections with thalamic neurons, which activate the motor cortex, facilitating movements. Again, in the indirect pathway, we have dopamine that gets involved. There is an inhibitory effect, inhibits the thalamic neurons, inhibit motor cortex, and inhibition movements. So what happens? So there is a loss of balance in the striatum. There is a degeneration of the GABAergic neurons. There's an imbalance of dopa and GABA, and that will cause the abnormal movements. What are the symptoms? So Huntington chorea is one of the major symptoms. So the symptoms include the abrupt, involuntary dance-like movements involving the face, trunk, and extremities. There is ethetosis, which is the involuntary writhing movements involving hands and finger. And then we have the cognitive or behavioral symptoms. There's memory deficit, there's the diminished decision making, affective disturbance like personality changes, mood disturbance, dementia, psychosis. So these are one of the major symptoms that you see. So we're going to discuss the both of these disease further down when we have the topic of aging. We're also going to discuss that when we're going to start our revision for the USMLE CK. So thank you so much. And in the next presentation, I'm going to discuss the cerebellum, and that presentation is coming soon. Thank you so much for supporting, and I'll see you soon. Have a good day.